Okay, let's get started. Hello, everyone. Welcome to the Environmental Research webinar. I am Sungkyun Park, faculty member of Epidemiology and Environmental Health Sciences. So this seminar series is sponsored by the Michigan Life Stage Environmental Exposures and Disease Center, M-LEEaD. So today we have one of our center scientists as a speaker, Dr. Sara Stein. So Dr. Stein is a research assistant professor in the Department of Health Behavior and Health Equity here in University of Michigan School of Public Health. As a clinical scientist, so Dr. Stein examines the transmission of the effects of trauma from mother to child prenatally, postnatally, and when women are parenting young children. The goal is to reduce these effects in families by improving interventions for trauma-exposed women as they become parents. Ultimately, Dr. Stein seeks to create treatments that target intergenerational transmission and that are responsive to community needs. She centers her work in understanding how mothers are shaped by the social structures in which they are embedded. The research is grounded in her clinical experience with structurally disadvantaged women with complex trauma histories. Dr. Stein's clinical work as a fully licensed clinical social worker integrates evidence-based approaches and translates research into trauma-informed public scholarship and clinical tools for adults, children, and clinicians. So Dr. Stein's talk is an application of her theory into environmental health, which sounds very interesting. So please join me in welcoming Dr. Stein. Oh, Sarah, yeah, please unmute.
I am muted. Sorry about that. Good afternoon, everybody, and thank you, Dr. Park, for the introduction. Yes, my name is Sara Stein, and I use she/her pronouns. And I'm delighted to be here today to talk about the double hit to health, the mixture effects of early life heavy metal and cycle exposure on health, different health outcomes. Today, I'm going to be talking a bit about how I got into this work, kind of giving this kind of broad introduction, and then my goals as a center scientist here in the M-LEEaD Center, which I'm very excited to and honored to be a part of. And then I'll be presenting on 2 different studies that I am working on with collaborators, and then I'll be talking about some of our future directions. I hope to be able to keep this to about 30-ish minutes, and then I look forward to taking any questions that you all might have. Okay, without further ado, I will go ahead and get started. So my work generally focuses on supporting adult women in order to improve health outcomes for children. As Dr. Park already kind of gave a little introduction, through my research, I seek to answer the question of how do we stop the intergenerational transmission of the health effects of trauma. I study this in mothers and children from pregnancy all the way through early childhood, because this is both a period of time of both immense vulnerability, but also opportunity due to rapidly developing brain, stress response, and attachment systems. My work bridges both basic and translational science. On the translational side, I identify pathways of transmission focusing on women's health and parenting. And on the translational side, I use these findings to improve and develop interventions to stop intergenerational transmission. The bulk of my work over the last 10 years has focused on the impacts of trauma and psychosocial adversity on women's health, women and children's health, and parenting. What we know is that estimates are that about a quarter of the population has experienced 4 or more adverse childhood events, and that this number is higher in women and individuals of childbearing age. Some of the adverse events that I have focused on in my work have been things like childhood maltreatment, including physical, sexual, and emotional maltreatment and neglect, as well as intimate partner violence or dating violence or domestic violence, which are all synonyms. So looking at violence, interpersonal violence in adulthood. Another really important piece of my work, another type of psychosocial adversity or trauma that I focused on, is that of systemic inequities that overwhelm a person's nervous system capacity to cope, such as experiences of racism. And what we know is that psychosocial adversity or trauma, I'm kind of using these terms interchangeably right now, and I'm happy to talk about some of the nuances later in the question and answers, especially in early life, have detrimental and long term health impacts. They have the outcomes, these are outcomes that we generally care about: birth outcomes, neurodevelopment outcomes, chronic disease like asthma, cardiovascular impacts, severe mental health problems, including depression, anxiety, and impact physical health as well as other physical health outcomes. But something that is largely unexamined in the trauma research and the trauma literature are environmental exposures. So when we think about systemic inequities, I have been looking at racism, access to housing, access to employment, but the trauma research is really not looked at environmental exposures. As this group probably well knows, there are multiple types and sources of environmental exposures, including heavy metals, pesticides, PFAS, air pollution, microplastics, BPA, among others. And we also know that children are especially vulnerable to environmental exposures. And why is this? Children, as many of us know, will spend a lot of time putting their hands in their mouth. So they are taking in things in our environment more readily and more often than adults. They also have increased absorption rates compared to adults. And as I said earlier, we know that these are sensitive and critical windows of development for the developing brain, the nervous system, different physiological things that are happening during this time. And finally, we also know that environmental exposures, especially in early life, also impact health and have actually similar outcomes to the ones that we were seeing with trauma. So the environmental exposures are linked to negative birth outcomes, problems in neurodevelopment, chronic disease, mental health, and physical activity. So we know that psychosocial adversity or trauma and environmental exposures have these shared health impacts. We also know that those that are living with high psychosocial adversity often are the same communities that are disproportionately burdened by environmental toxicants. And we also know that kids do not just have one exposure adversity in their life, right? So it's not that kids are just exposed to um, one type of heavy metal, but that if you're in a space and in a community that are burdened by environmental toxicants, you're going to have many different toxicant exposures. And what this data is also showing, or this evidence is also showing, is that you're not only going to have these heavy metal or toxicant exposures, but you're also going to be having, likely, there's this overlap with having psychosocial adversity. So, what I am interested in, have been collaborating with some environmental neuroscientists, is this idea of a double hit. It really brings us to this idea of a double or two hit models of stress, which postulate that repeated exposures to stressors can result in physiological changes to our stress system, including stress system alterations, neurochemical effects, which influence development and health trajectories. For example, experiencing a stressor like poor nutrition and important life stages like the prenatal period, and then again in childhood, can result in double hits on stress response systems that will impact developmental trajectories. But what I'm interested in looking at is this double hit of experiencing both a heavy metal stressor or a toxicant stressor in childhood, as well as experiencing a psychosocial or trauma exposure in childhood, and seeing how these things combine to impact health risk later in life. So, given that they co-occur and that we know that children are experiencing multiple exposures at one time, we know that multiple environmental and psychosocial exposures should be considered together. Heavy metals and psychosocial factors may interact and have joint impacts on health outcomes, and their combination could compound risk for biological and physiological adaptations in adolescents, which influences a life course health trajectory. So we know that both environmental and psychosocial exposures in early life influence long-term health. Oops. And how might this happen? Heavy metal exposures and exposures in early childhood could influence neurodevelopment and physical health in adolescence, as well as our stress biology via the HPH axis, epigenetics, and immune processes. And that we postulate that then this may affect cognition, mental health, and physical health in adulthood. Now, where is the problem? The problem is that these areas of work have really existed in silos. So there, as we know, there's a huge toxicant literature that has examined the different health impacts of being exposed to different toxicants in different developmental periods, very often in childhood and prenatally. And the trauma literature there's a huge body of work that has examined this in the effects of experiencing trauma on health impacts throughout the life course. And what my work and my work in collaboration with Dr. Olivia Halabicki postulates is coming together as an interdisciplinary team to combine our two areas of expertise, mine in trauma and hers in environmental toxicants, to come together to be able to tackle these issues together and to really be able to model the real world impact of being exposed to both toxicants and trauma exposure on our health long term. I was honored to be selected as a center scientist, the M-LEEaD center scientist, one of the two M-LEEaD center scientists this year. And my training goals are to strengthen my area of work in the area of the toxicant world, in order to be able to capitalize on this interdisciplinary work that I am doing. So today, as you'll be seeing the work that I'm doing, you'll be seeing that I'm focusing on three training goals. The first goal is to strengthen my expertise in environmental toxicants and stress-related biological mechanisms. As I noted earlier, my expertise is in trauma exposure and the health impacts of that. So this is a newer area for me. My second training goal is to advance my statistical expertise in modeling early life exposure mixtures and physiological stress mechanisms. And so there are some as many of you are likely familiar, there are methodologies in modeling mixtures that I'm hoping to get, like G computation and BKMR, that I'm hoping to get further training throughout this year. And I'll be talking about one of the studies that I'm using those in currently. And finally, and a great part of doing this webinar as well, my goal is to build interdisciplinary leadership, cohort development expertise, and a capacity for public health translation of findings. So you all will tell me how I was able to translate some of my work into hopefully easy, digestible, understandable findings today in the presentation. So without further ado, I am going to go ahead and just talk about two studies that I am working on in collaboration with Dr. Halabicki. And I will walk you through them, and I look forward to answering any questions at the end of the presentation. So this first study is a systematic review and a state of the science on the joint effects of toxicant and psychosocial exposure. Because I've already given you an overview of the method, the kind of theoretical framing, and the issue of the problem, I'm going to kind of just jump in straight to the purpose of this review. The purpose of this review was to synthesize existing evidence of compounding effects of toxicants and psychosocial co-exposure or mixture effects on health outcomes. For the purpose of this presentation, I will be using joint exposures, co-exposure, and mixtures as and interchangeably. And the second purpose of the review is to characterize three different things: the types of environmental and psychosocial exposures, as well as health outcomes tested, the developmental timing of the tested exposures and outcomes. So when in development were the exposures assessed, and when were the outcomes assessed? And we're hoping to characterize a focus on the compounding risks or protection of these mixtures. So we did a comprehensive search using toxicant exposure and psychosocial exposures using PubMed, PsycInfo, Embase, and Web of Science. We followed the PRISMA guidelines, 2 reviewers, first by title and abstract, and then we did full text review. In order to be included in our study, the study had to be focused on toxicants, have a toxicant and a psychosocial exposure. And the study had to test for an interaction or look at stratification or mixture effects on health outcomes. We excluded animal models, mediation testing that did not include some sort of interaction to see if there's an exacerbating effect of the combination of the 2 exposures, and neurocognitive outcomes. We were just focused on health outcomes. We did not have any restrictions on the population by age or geography or any other factors. Let me just quickly show you our the PRISM diagram. Just to quickly note, we screened about 5,000 studies, and almost 58 studies were included in our final body of studies. So I'm going to just quickly go over the results. By landslide, the highest number, the toxicant type that was found in highest number was air pollution. And SES was by far the psychosocial exposure that was most assessed. It's important to note that these studies may have, many of the studies examined several different types of psychosocial exposures, or several different types and different occasions of toxicant exposure. So these numbers will not actually add up to 58. The different types of health outcomes, there were a number of different types of health outcomes. The 2 highest were birth outcomes, so looking at low birth weight, amongst other things, and respiratory outcomes like asthma. But cardiovascular and mental health outcomes were also some of the more popular outcomes that were assessed in this body of literature. And by far, the prenatal time that was the focus for the timing of the exposures was the prenatal period. This was very much true of the toxicant exposures, as well as a lot of the psychosocial exposures. So we saw that by and large, SES is the largest psychosocial, or socioeconomic status is the largest psychosocial exposure that we were looking at. So it was very common that the studies would be looking at, for example, heavy metal exposure or air pollution during pregnancy, and be assessing SES at that same time. The developmental timing of the outcomes was much more varied. Certainly, the newborn and childhood period were the most frequent, but adolescence and adulthood, we also saw a number of studies looking at outcomes. And then we also assessed for the interaction or mixture effects of that were assessed. And what we saw of the 58 studies, 50 of them found significant interaction effects. Mostly finding, most of the studies were focused on toxicant exposure and whether a psychosocial exposure exacerbated these effects. And what was found is that kind of almost universally, the psychosocial exposure exacerbated the effect of the toxicant exposure on whatever health outcome was being assessed in the study. So a few different things we found: a greater level of prenatal exposure to PHA, which is predicted lower odds ratio for infant reactivity at 4 months only when maternal stress was high. Associations were also positive for persistent respiratory symptoms, asthma ever with current respiratory systems among children with a history of stressful family events, but not among children without a history of stressful events. And we observed an interaction between the overall mixture and PNSDI score, which is prenatal social disadvantage index, in which a quartile increase in the mixture was associated with greater risk of depression symptoms for the low group, but not for the high group. And one of the big findings that we also have here that's actually not represented on the slide is that of the 58 studies, only 3 of them used mixture effects, mixture methods, so something like G computation or BKMR to find these results, or to assess for their analyses. And the majority of the studies used either a stratified analysis or an interaction. Let me do a quick discussion of our findings. The overall findings are that the heterogeneity of environmental and psychosocial exposure assessment, health outcomes, and methods to test the joint impacts makes it difficult to draw conclusions. So we were looking at many different health outcomes, many different environmental and psychosocial exposures. So we can really talk about kind of what the body of literature is looking at and where our next steps are, rather than drawing conclusions about what this means for a specific health outcome or for double hit outcomes. Oops. But there does appear to be evidence for a stronger effect of toxicants on health outcomes for persons of low SES experiencing individual or home level, persons of low SES, or those experiencing individual or home level stress. So one of the issues that we found in this literature is the issue of how we're conceptualizing these psychosocial exposures. So the significant findings for toxicant by the majority of the findings were focused on toxicant by non-specific SES measures, rather than looking at some of the psychosocial exposures that I look at in my work and that we know are really associated with long-term health outcomes. So SES and these non-specific SES measures like income or occupation, which often were at census tract level, are not a proxy for psychosocial stress or experiences. So this study really shows that future work needs to consider multiple psychosocial aspects at the individual and the neighborhood level, and combine it with this mixture approach. Studies largely focused on compounding risks instead of potential protection, and this helps us to understand who is at greater risk for adverse health outcomes. But few studies conceptualize a lack of risk as a sign of protection. And why does this matter? Protection is not the opposite of risk. And so in our future work and the work that Olivia and I are doing, true protective factors really need to be tested. And I'm happy to talk more about that. Timing of exposures and outcomes. The adolescent period is a far less studied period, especially when considering the health outcomes in that period of time. And why does this matter? We know that this matters because it's a critical time of development, which may set the trajectory for long-term health. And methodologies, as I already said, most or all studies included interactions between a toxicant and a psychosocial exposure, or they stratified analyses by psychosocial exposures. Only a few studies, as I mentioned before, only 3 used mixture approaches. And why does this matter? Single exposure models won't capture the whole picture. And if we're really trying, kind of the goal of our work is to be able to capture real world or to model what is happening in the real world, these approaches are not sufficient. Mixture approaches like GComp or BKMR may help identify unique and potentially nonlinear combination of exposures, which can increase risk or provide protection. So there's really a call here for interdisciplinary work, what Olivia and I are really excited to be doing. Um, NIEHS has called for work that has this interdisciplinary focus to reduce individual susceptibility for long-term health adverse health outcomes by identifying co-exposures and biological mediators. There's a very large focus on prenatal and early life exposures to both toxicant and non-toxicants. And developmental psychology or clinical psychology can really play a big role in this. So I am joining the toxicant world with this kind of developmental and clinical focus in order to be able to address this NIEHS priority. And just looking forward towards these protective factors, something that our pending R21 will hopefully be able to address. But protective psychosocial factors could be leveraged as interventions to mitigate harm even after a toxicant exposure has happened. So it's really an important area of research and area of research and a place for future policy development. So our research really needs to be focused on combinations of exposures, timing of experiences and exposures, and where targeted interventions could be most impactful. Oops. So unless we do this, environmental exposures and inequities will continue to impact child health. We need to be able to promote through understanding these mechanisms and these mixture effects, promote individual and family autonomy to protect child health and development. And now I'm going to quickly move on to our second study. And this study is using empirical data. So the first study was kind of looking broadly at the literature and understanding kind of where some of the gaps are when looking at these mixture effects. And the second study is looking at the interactive associations between prenatal metals and childhood socioeconomic position on adolescent allostatic load. This is work that is currently being led by Olivia Halibicki and myself, and is very much following, is in response to the the findings of the systematic review that I just presented. So, what is the purpose of this project that we are doing? It is to examine individual and mixture effects of prenatal exposures to heavy metals and socioeconomic position on adolescent allostatic load, which is an index of cumulative body burden of physiological stress, and well as of individual biomarkers. And we're going to be doing this, or we do this using data from the prospective birth cohort, the prospective birth cohort element, which I will talk about just now. So I have been delighted to be able to join Dr. Olivia Halibicki, as well as my mentor for the center scientist position, Dr. Karen Peterson, on the element study, which is a longitudinal birth cohort centered in Mexico City. This was started at Harvard University and then came to the University of Michigan School of Public Health, and it is a collaboration with several institutes and groups in Mexico City, including the National Institute of Public Health, the Centro Médico, the ABC hospital, as well as the Instituto Nacional de Periodontología. And there are many different researchers located across the United States, as well as in Mexico, who are contributing and who were the founders of this birth cohort. So this cohort, for those who are not familiar, is a birth cohort. Mothers, primarily during pregnancy or just after birth, were enrolled starting in 1994. And there's a current follow-up in 2025 that is looking at that is following the children of the mothers that were recruited starting in the 90s. about half of the cohort, the children that were born are female, and again, and the beauty of this cohort is that we have these maternal dyads. We have this kind of intergenerational piece to, or there is this intergenerational piece to it. So we have what happened in birth. There is an early childhood follow-up, a late childhood follow-up, an early adolescent follow-up, and again, now, as I mentioned, they are currently following up on early adults. The kids are between the ages of about 18 to 30 right now. Just after there were initially about just about 1,600 mother-child dyads. And there were about 525 adolescents that were assessed between 10 to 18 years in 2015. The projections for the 2025 data, which we will not be discussing in this study, are around 550 young adults. So let me quickly get into the methods. First, Dr. Halabicki developed a allostatic load index, which included 14 biomarkers from 4 body systems, including the cardiovascular system, metabolic system, our immune systems, and the neuroendocrine systems. And she created this using sample distribution methodology. We looked at metal exposures that were log blood and urinary metals were collected at that first time point, and then the SES or the socioeconomic position was assessed also in the prenatal period. And then we are looking at adolescence. We're looking at allostatic load. And the individual biomarkers. Covariates were age, sex, BMI, and pubertal age, specific gravity of the urinary measurements. And essentially what we did is we looked at linear regressions of both the toxicant exposures, the metal exposures, as well as the socioeconomic position. And then we looked, as many of the other studies did, in the systematic review of the interaction, or we did stratified analysis. But we also did mixture exposures to look at the mixture's impact on allostatic load. So given time, I'm going to go through this very quickly. I'm happy to chat further about this. But here are the sample. As you'll note, that the sample is quite young. The average age of the sample is quite young. And many of the adolescents were in early stage puberty. It was also kind of fairly split between high and low status SCS. So here are the linear models. The first predicting allostatic load, then the next 4 you'll see are predicting individual biomarkers. And what we'll see is that antimony and manganese and AMAI, lead, and arsenic are all significant predictors of the different either allostatic load or the different individual bio outcomes. And then here we'll see in yellow that these were marginally significant. And just looking at the marginal mixture effects of metals on allostatic load. And then we have some significant mixture effects on some of the individual biomarkers. For insulin-like growth factor, we see that here we see this. We also have some marginal effects on cortisol. And then we did some linear models stratifying by SES. And we saw that it was really that lead was a driver in allostatic load, and for systolic blood pressure for those individuals in the low SES group, and that it was cobalt was a driver in the high AMA group, in the high SES group. We also had some marginal effects here, which I'll be talking about. I'm going to quickly go through my discussion here. allostatic load was largely non-significant in individual and mixture models, but individuals' biomarkers were significant. And why might this be? Allostatic load, the allostatic load takes time to develop. Allostatic load or overload takes time to develops over time after repeated hits and physiological adaptations and impacts to the entire body. So adolescents may be too early to see effects on allostatic load. This is a very young sample, like I mentioned. The mean age is 12.8 years. And what we did see is that the individual biomarkers within the allostatic load index had significant or marginal associations with heavy metals and SES in a linear and the mixture models. These individual biomarkers are more upstream, hormonal or neuroendocrine biomarkers, and may be indicative of later development of allostatic load. Low SES appeared to increase the effects of metals on allostatic load and individual biomarkers. So for those in the low SES groups, the adverse effects of lead on allostatic load was significant, though it was not for the high group. And what do we think SCP captures here? It's a limitation here of secondary data. So SCP stands for socioeconomic position. It's not a good proxy for stress or well-being. So we need to continue to develop robust measures of psychosocial exposures and to be included in environmental health research. And I'm just going to, given the time, I'm going to, and we can use, if we do this, we can use these findings to develop individualized interventions to mitigate developmental harm even after intervention. So that's really what we're going to be focusing on. So what are my next steps as a center scientist? I'm currently working on another study that parallels the study that I just presented that is going to be looking at adolescent oxidative stress. So these mixture double hits, the effects of psychosocial childhood socioeconomic position and prenatal metals on oxidative stress in adolescents using the element cohort. And the beauty of this work is that oxidative stress is probably more of an we're likely to see more upstream effects given that the mitochondrial changes that are that is oxidative stress. So we are hopeful that we may see this, that we may see more signal here as we begin to look at this, and that is imminent. We are working on the analyses right now. And then our larger body of work is we are working on expanding the psychosocial data in the element cohort. We are, thanks to a currently funded U24, which Dr. Peterson and Dr. Song are co-PIs or MPIs of. We are going to be able to collect some retrospective data on psychosocial exposures, particularly the adverse childhood experiences that the now young adults experienced when they were children. So this is really going to allow us to expand our capacity to look at these mixture effects. As well as Dr. Halabicki also has some recent K99 R00 funding that is also going to allow us to look at some protective factors retrospectively assessed as well in the young adults, but protective factors like benevolent childhood experiences. And we're going to be able to look at those in combination with the heavy metal mixtures, heavy metal exposures that are already well characterized in this sample, and be able to look at the effects, the potential associations with young adult mental health, which are understudied in the metal world, not in the psychosocial exposure world, but in the metal world, and certainly in the mixture world. The effects on mental health are, some studies have looked at it, but it's just a handful of studies. And this work that we're doing is also going to be looking at the potential mechanisms for this positive association, including allostatic load and oxidative stress. And we have an R21 that is currently pending. We'll see what happens with it. That will allow us to map all of this out. So I know I'm a bit over time. I want to thank you so much for your attention. I hope that this gave you a brief overview of the kind of the directions. I'd like to thank Dr. Karen Peterson, who is my mentor here as a center scientist, Dr. Olivia Halabicki here on the left, who is who has spearheaded with me this interdisciplinary work, and my other mentor, Dr. Allison Miller. And with that, I will end. Thank you.
Great. Yeah, this is really interesting topic, and I agree that so it is very important to to collaborate these two silos between environmental health community and then this health behavior, health equity, social factor, social science, and I think this is a very underdeveloped, understudied area. But there is a lot of potential opportunity to address this potential public health issue of this early life exposure, environmental exposure, and how this early life environmental exposure can be even, the amplified or can be mitigated depending on how the social stressor can be, like a social psychosocial stress management can play a role. And so I'm very glad to see this work, and also your future plan of focusing more on the like a protective factor.
Yeah.
We look forward to additional work that you can the develop potential intervention strategy to mitigate this environmental exposure effect. So we have a few questions, and so let me read some question, and you can also see the if you open
Sure.
the Q&A. And so the first question and the fourth question, I believe you can address at the same time. But the first
Okay.
question, so could you give an example of a health outcome that came about due to early life environmental exposure? What do you know about the mechanism behind the interaction between early life environmental stressor and long-term health outcome? And the fourth question is also, can you explain why psychosocial factor can compound or modify the effect of metal exposure on health outcome?
Yeah. Can you confirm the? So I'm just making sure I can answer both. So what can give us the health outcome and what we need? So I think one of the things in the and I hope I'm going to be answering this right, but one of the things that has been shown is that, and at least in animal models, that individuals that have been exposed to, or rats that have been exposed to lead, let me start over. There's prior work to show that exposure to some toxicants like lead exposure does not predict universally bad outcomes necessarily. That things like specifically education can really buffer some of these or lead to differential health outcomes. And so I think what we are attempting to do is to say, well, why is this? What about the psychosocial exposure is providing some sort of protection physiologically when kind of there has been this universal toxicant exposure. So I think we are very much the point of our work is to begin to be able to look at the mechanisms of these interactions, and we are postulating that these kind of upstream things that we are hoping to see, or that we're postulating we'll see in adolescence, will be some of the mechanisms that will be able to explain some of these effects on the long-term health outcomes. But I think the answer is that we don't yet know. I'm happy to be in conversation if others have some other information to provide. But I believe that is very much the point of this work, is to understand the mechanisms in order so that we can develop interventions to target those mechanisms, so that and that's why we're looking at some of these beneficial, these protective factors, because if there are, if education or if X, Y, and Z can serve as a protective factor, then we can get in even after an environmental exposure has happened to help mitigate the effects on health for kiddos at different stages.
Mm. If I add a little bit more, then so I think the main reason why your study focus on allostatic load is because the allostatic load or chronic stress response is one of the
Correct.
very important like a mechanism behind how this chemical exposure and then the psychosocial stress they play together, and then through this the HPA axis, and so that's
Yes.
you focus on that outcome. Although as you mentioned that the among adolescent, so
Yes.
the change in allostatic load maybe is too early to observe any change in this one. So that could
Yeah.
be important, the mechanism we can address. And so also the Dr. Devon Payne Sturge also leave up the comment, but her study of this environmental exposure and then psychosocial stress at the neighborhood level and the individual level, and how this so segregation, to perpetuate this chemical exposure and then the psychosocial stressor play a role. And so I also recommend
Yes.
her work. And so her like a comment is also like a thank you for sharing the result. And then
Yep.
the Dr. Payne Sturge also did similar work. So you can find her scope.
Yes.
Yeah.
That's wonderful. Thank you for sharing,
Okay.
Dr. Payne Sturge. Yeah.
And then another question, so why neurocognition outcome are excluded as a part of health outcome? And I think this is part of your systematic review? I'm not
Yes.
sure.
Yes.
Yeah.
Correct. Because we are both interested. I think it was a largely because we are both interested in mental health and other physical health outcomes, and the neurocognitive outcomes get into many different spaces, including obviously dementia and different spaces that were just not the focus and are not as tightly linked to trauma exposure. And so we kind of stayed in a lane that is more tightly linked to trauma exposure, I would say, the health outcomes associated with trauma exposure. So it was a decision as a research group. But yes, but also it seems like there is a lovely already a systematic review that is looking at neurodevelopmental outcomes.
Okay, so before we end today's webinar, and I want to ask, so even I am very interested in the psychosocial stress and how it is related to or interact with the environmental toxin exposure. But the challenge is how to measure
Yes.
the psychosocial stress, right? And
Yes.
the one way is using the like a questionnaire to and then based on the record, and as part of your allostatic load, you also measure more objective stress response as like a cortisol as a stress response
Yes.
biomarker. And but either way, so there's a limitation of capturing this psychosocial stress and evaluate this interaction. So I'm just curious. So what is your like a strategy to pursue this direction, and then what is the best way to measure psychosocial stress?
Yeah, that's a great question. And in fact, in our review of R21, that has been something that's been brought up by multiple reviewers. That there is a retrospective reporting on psychosocial stress, and I'm not sure that there is a way around that. I mean, certainly there are like objective measures, like using CPS records or like public records or hospital records to assess psychosocial stressors. But I mean, that is something that you can do, and that's a kind of more of like a quote unquote objective, like real time assessment of a psychosocial stressor, but really does not get into the subjective experience for the individual of the stressor, which is arguably potentially more important than the just the kind of like the objective, did this happen or did this not happen? we can quibble about that, but that is one thing. And also, we are not through some of these other kind of more objective assessments, we are not getting at some of the more nuanced psychosocial exposures that we know are really impactful for health, like emotional neglect, like different forms of emotional abuse, which would not bring somebody to a hospital, that would not necessarily elicit a CPS call and a CPS record that would be part of a record that we could evaluate. And so I think the answers are that our systems for capturing, like objectively capturing some of these of these exposures are quite limited. And that it is not only just like, did this happen, but the impact and whether you consider it like important enough to to endorse it, that I think actually matters for health as well. But again, these are things that these are nuances that we need to be working with. But it's a very good point, and it is a challenge as we are going forth with this work. So thank you for bringing that up, Dr. Park.
Okay, all right. Yeah, so unfortunately, we have, ended this session today. And so thank you for coming to this webinar. So next week, we will have our first residents and researcher webinar about learning and living with wildfire smoke. So please stay tuned and then join again next week. So thank you very much.
Thank you, everybody. Thank you, Dr. Park.